Vegetative propagation by leaf cutting produces plants genetically identical to the parent. Snake plant, Sansevieria, is suitable for this method due to its thick leaves that store nutrients and water. This experiment examines the effects of leaf cutting length and polarity on propagation success to identify optimum conditions.
The leaf cuttings were cut into lengths of 3.5 cm, 7 cm, and 14 cm. The base of the leaf cuttings was cut at a slant into a pyramid shape. For experiment 2, the other two leaf cuttings were prepared. One 14 cm leaf cutting was cultured in an upright position, while the other leaf cutting was cultured in a bottom-up position. The end of each leaf cutting that was to be cultured in water was cut at a slant into a pyramid shape. All of the cuttings were placed into a cup containing water with the cut edge submerged. The experiment was performed in duplicates. All of the cups were placed in the incubator. The cuttings were observed daily, and the dates on which roots and shoots appeared were noted.
.5 cm leaf cuttings formed tiny short roots. Root initiation requires less energy and fewer stored reserves compared to shoot formation. Short cuttings still retained active meristematic cells at the cut surface capable of forming adventitious roots. The reduced tissue mass lowered respiratory demand, allowing the cutting to survive long enough for root initiation. 7 cm leaf cuttings failed to form roots because larger cuttings had higher metabolic and respiratory demand. In water culture, oxygen availability at the cut surface may have been limited, causing tissue stress or decay before root initiation. Stored carbohydrates were consumed for maintenance respiration rather than root differentiation. This resulted in tissue deterioration before roots could develop. 14 cm leaf cuttings exhibited the earliest and most vigorous root growth. Roots consistently emerged from the basal end submerged in water. This length was the most effective for roots formation. 14 cm upright, normal orientation, roots developed from the basal end placed in water. Growth followed the natural polarity of the leaf. Auxin accumulation at the basal end stimulated root formation. Cytokinin activity favored shoot initiation at the upper region. 14 cm bottom up, inverted orientation, root growth was still observed at the end submerged in water, even though it was originally the apical end. However, root initiation was slower and less uniform compared to upright cuttings. Shoot emergence was delayed or absent. This indicates that although snake plant has strong regenerative ability, natural polarity was disrupted, affecting hormone distribution.
Optimum condition. I. Leaf cutting propagation of snake plant. Leaf cutting length, 14 cm. Longer leaf cuttings produced earlier and stronger root and shoot formation compared to shorter cuttings, as they contain higher stored carbohydrates and water to support regeneration. Correct polarity, upright orientation, cuttings placed with the basal end downward successfully formed roots and shoots, while inverted cuttings failed, showing that polarity is essential for proper auxin movement. Next, for optimum condition, auxin transport. Correct polarity allows auxin to accumulate at the basal end of the cutting, which stimulates adventitious root initiation. Moisture condition. Adequate moisture prevented desiccation of the cutting and supported continuous metabolic activity during root development. Lastly, for optimum condition, light condition. Indirect light reduced stress and supported healthy shoot development without excessive water loss. Overall optimum condition. A 14 cm leaf cutting, placed upright with correct polarity, maintained under adequate moisture and indirect light provides the most favorable condition for successful snake plant propagation.